Welcome back to Neutralizing COVID, the monoclonal antibody story, the podcast where we unpack the science, strategy, and real world impact of monoclonal antibodies in the fight against COVID 19. I'm your host, Dr. Shikha Agrawal. In this series, we have been diving deep into how monoclonal antibodies have played a role in the COVID 19 response. In today's episode, we will go behind the scenes to explore manufacturing and logistics challenges in manufacturing challenges and cold chain logistics in monoclonal antibody deployment. To bring clarity and context to today's discussion, thrilled to have our nanomedicine researcher, Dr. Aiswarya Chaudhuri, here with us again. Thank you for joining us, Dr. Aiswarya.
Thanks for having me, Dr. Shikha. I'm glad we are spotlighting this part of the story. It's often behind the scenes, but absolutely critical.
To start us off, could you walk us through how monoclonal antibodies are manufactured at scale, particularly in the context of therapeutic use?
Sure. Monoclonal antibodies are typically produced using genetically engineered mammalian cells, most commonly Chinese hamster ovary or CHO cells. These cells are cultured in bioreactors, which are large, sterile tanks, where they grow under tightly controlled conditions. The process involves upstream production, where the cells produce the antibodies, and the downstream purification, which removes the impurities to ensure a safe, consistent product. The entire process must meet the strict GMP, that is the good manufacturing practice standards, which are essential for safety and efficacy.
That sounds incredibly complex. What are some of the major manufacturing challenges unique to monoclonal antibodies?
One major challenge is scale. These are complex proteins, not small molecules. So production can't just be wrapped up overnight. It takes weeks to grow the cells and complete the purification. Another challenge is yield, the efficiency with which the cells produce the antibody. Improving yield without compromising quality is an ongoing focus in manufacturing science. And then there's consistency. Every batch must be nearly identical to meet the regulatory requirements, which means even small process changes need extensive validation.
So, once the product is manufactured, getting it to the patient poses a new set of hurdles. Let's talk about cold chain logistics. What makes this part of journey so demanding?
Monoclonal antibodies are temperature sensitive biologics. Most need to be stored between 2 degrees Celsius and 8 degrees Celsius and must be protected from temperature fluctuations throughout the supply chain. That is from the manufacturing site through transport to hospital or clinical storage. If the cold chain is broken, the protein structure can degrade, rendering the product ineffective and unsafe. That means investing in the refrigerated transport, temperature monitors, and trained personnel at every step. This becomes particularly difficult in low resource or remote settings where cold storage infrastructure may be limited or unreliable.
I appreciate that insight. Have we seen real world consequences when cold chain logistics fail?
Yes, unfortunately, there have been cases globally where temperature exertions led to product spoilage or the need to discard the doses. In a public health emergency, that represents a serious setback, both in terms of cost and lost time. Maintaining cold chain integrity is critical not just for safety, but for public confidence in biologic therapies.
Are there innovations on the horizon to help overcome some of these limitations?
Definitely. There's active research into formulation science, for example, creating monoclonal antibodies that are more stable at higher temperatures or dry powder formulations that don't require the cold storage. There are also efforts to decentralize the manufacturing with smaller modular facilities closer to end users. And digital tracking tools now allow for real-time temperature monitoring along the distribution routes, improving visibility and rapid response.
Great overview, Dr. Aishwarya. Do these manufacturing and logistical challenges impact access, particularly in low and middle income countries?
Absolutely. High production costs, limited manufacturing capacity, and a low chain requirements all contribute to inequitable access. Even when the intellectual property is shared, the infrastructure to produce and distribute the monoclonal antibodies may not be in place. Addressing these barriers is key to making monoclonal antibodies a part of the global solution, not just a tool for high income settings.
As we look ahead, what's needed to strengthen the monoclonal antibody supply chain for future pandemics?
We need investments in local biomanufacturing, global supply chain coordination, and resilient cold storage systems. We also need regulatory pathways that allow for rapid scale up without compromising the quality. Preparing now will ensure that monoclonal antibodies or similar biologics can be deployed quickly and inequitably in the next crisis.
A fascinating and important conversation. Dr. Aishwarya, thank you for joining us and shedding light on the science behind the scenes.
Thanks a ton, Dr. Shikha. It's been great.
That's it for this episode of Neutralizing COVID, the monoclonal antibody story. Manufacturing and logistics may not get headlines, but they're foundational to the therapies we rely on. To our wonderful audience, thank you for joining us for another deep dive. And hey, if you like this episode, why not spread the word? Share it with a friend or on your socials. Until next time, take care and keep exploring science.